Okay, hello. This is now the third topic for midterms. So, amino acids and proteins, specifically the biochemistry and clinical applications of this one. For the laboratory methods and processing, it will be discussed in the next topic. Okay, same with your other biomolecules. So, between the two, we're going to discuss first your amino acids because based from your biochemistry, if you can remember, your amino acids are the building blocks of a more complex compound, which is proteins. Okay, so let's review first the biochemistry of amino acids. So, by the way, before I continue, I would just like to inform you that this topic will be divided into two video lectures because I want to um, not make it look like very long. No, this is a very long topic kasi. So the first video lecture, which is this one, will focus on the amino acids, and then the second video lecture will be about proteins. So let's have amino acids now. So remember, amino acids, they have parts. Okay, so you have here the amino group, and then you have here the carboxylic acid group. So they are named as such because of the functional group that they are. Okay, so that's for the two. Then you have the side chain or the R group. So the side chain is the hydrocarbon part of the amino acid. Then you have here a hydrogen atom. Then the carbon at the center of the amino group, carboxylic acid group, the side chain. The carbon at the center is known as the alpha carbon. So if you can remember or if you recall, it is named as your alpha carbon because it is the first carbon atom that is attached to a functional group, and even the side chains are bonded to this. Okay? So also talking about amino acids, di ba? There are a total of 22 amino acids. Okay? So 20 lang po ang pinakita dito kasi yun yung established dati pa. Okay? But if you're going to look at the book, there are actually 22. So ang dalawa po na naidagdag dapat dito ay si selenocysteine and your pyrrolysine. Okay? So talking about amino acids as well, no, we also need to remember, no, that your amino acids can be classified according to the polarity of their side chains. So if you're going to classify them according to the polarity of their side chains, you have now this what we call non-polar hydrophobic, the polar uncharged, the polar acidic, and the polar basic. Again, they're classified into four because of the polarity of their side chains. Okay, so for the non-polar hydrophobic, di ba remember po, yung R groups nila, mostly, no, ang makikita nyo lang talaga dyan is hydrogen and carbon. No? So may iba nga na purely hydrocarbon lang talaga siya. No? Or, yeah. Then, for the polar uncharged, no, usually ang makikita nyo po sa R group ng polar uncharged na amino acids sa side chain nila, no, is usually makikita nyo dyan alcohol, phenol, thiol, or amide. Then for the polar acidic naman po, dalawa lang yan sila. You have your aspartic acid and your glutamate or your glutamic acid. So kung titingnan nyo po yung kanilang side chain, no, hindi siya makita dito. No? But supposedly, dapat itong oxygen dito, no, meron yan siyang katabi na negative charge. So yung negative charge na katabi ng oxygen dito, no, yun yung isa sa mga palatandaan natin, no, na yung ating structural formula, no, yung structural formula ng amino acid, okay, describes the amino acid as polar acidic. And then for polar basic naman po, kung titingnan yung side chain niya, instead of seeing negative charge, ang makikita po dito ay positive charge, no, sa kanyang side chains. Okay? Then, aside from classifying our amino acids according to the polarity of the side chains, you can also classify amino acids based on being essential or non-essential. Okay? So, in other words, based on the nutritional requirement of the amino acids. So if we say that the amino acid is essential, it means that it is required in our diet. No? However, even though it is required in our diet, your essential amino acids cannot be synthesized by the body, correct? No? And then these are your essential amino acids. Actually, for you to easily remember your essential amino acids, I actually made a mnemonic for this one. So you have here PVT Tim Hall. No, PVT Tim Hall. Okay? So that's my mnemonic for essential amino acids. No? Yung non-essential amino acids naman po, they are not required in the diet, but they can be synthesized by the body. So ang style po natin dito para madali ninyong ma-recall, no? I-remember niyo lang po yung mnemonic for essential amino acid na PVT Tim Hall, no? Para ma-memorize niyo yung essential amino acids. And then yung mga hindi niyo na na-memorize, no? Yung hindi kasali sa PVT Tim Hall, automatically you can categorize them as non-essential amino acids. Okay? Now, in your Bishop book, each of these amino acids is discussed. I don't know if your teacher in biochemistry discussed each of the amino acid, but to be safe, no, we will go back to the descriptions of each of these amino acids, but I will not elaborate every amino acid. I will not discuss thoroughly every amino acid because you can actually read them, no, in your book. Okay, so we will go through with each of the amino acids, but what do you call this? I'm just going to emphasize or elaborate some details lang po. Okay? So let's begin with your arginine. So what I would just like to discuss about arginine here, no, is that it is required for the generation of urea and synthesis of creatine. I hope you're still familiar with urea. Diba? Urea is the less toxic form produced from the ammonia metabolized in the urea cycle, correct? Okay, so yan yan, no, si urea. And then arginine also is required for the generation, or rather for the synthesis of creatine. Creatine, this substance or this chemical here, this compound here, no, is actually involved, no, in the supply of energy for muscle contraction, no. If we talk about supplying energy for muscle contraction, I would just like to also clarify, no, na hindi lang po yan siya creatine, but actually mismong compound na nire-refer dito ay si creatine phosphate. Okay? Si creatine phosphate. Okay? Pero ang gusto lang sabihin ng slide na ito is yung creatine part ng creatine phosphate, no? Pinoproduce siya or sinisynthesize siya with your arginine. Okay? And then, additional information lang din po, if we're going to break down creatine, you can now have this what we call creatinine. Okay? You will discuss more, or rather, we will discuss more about creatinine when we arrive to the NPN, yung non-protein nitrogenous substances, no, sa finals. Okay? Finals, yun siya na topic. Okay? The second amino acid is histidine. So, this is a direct precursor of histamine. So, madali lang po i-associate yung dalawa kasi same yung first four letters niya, no? Hist. Okay. By the way, before I proceed, I would also like to remind you, okay, na for every amino acid, aside from their descriptions, aside from studying their descriptions, you also need to remember the three-letter abbreviations of each amino acid. No need to remember the one letter na. Okay? Ang importante sa atin is yung three-letter abbreviation. But if you want to also study, no, memorize the one-letter abbreviation, that will also be good. Okay? Pero ang importante dito ay yung three-letter abbreviation and the descriptions. Okay? So that's for histidine. That's what I would like to talk about for histidine. For isoleucine, so this one, again, abbreviation, three-letter abbreviation. Don't forget about that one. So this one is needed for hemoglobin formation. Then you also have leucine, which is used for the optimal growth of infants and for nitrogen balance in adults. Then we also have lysine. So this one, no, this one is involved in the production of antibodies or your immunoglobulins, as well as no, one of its beneficial aspect is it can lower triglyceride levels. Also, it helps in the absorption of calcium and formation of collagen. Okay, then we have methionine. So this one should be very familiar to you. Why? Because remember, your methionine helps initiate translation of messenger RNA. In other words, diba, this one is the first amino acid that is encoded no, during translation. And if this amino acid is not encoded, then translation cannot proceed. Why? Because what is the codon that encodes methionine? It's actually the start codon for translation. No? Yung ating AUG, yung ating start codon. So your methionine is also a source of sulfur, and this one no, is justified by the structural formula here. No? If you can see, in the side chain of your methionine, you will see sulfur. So definitely, methionine here can be a source of sulfur. Okay, and what is the role of sulfur in the body? So the role of sulfur in the body is that it is for the normal metabolism and growth of the body. Okay, and also, no, FYI lang po, your methionine prevents brittle hair. Okay, so yung mga may mga problema sa buhok dyan, no? Malay nyo, methionine ang problema sa inyo. No? Kulang ng methionine. Okay? Next po, phenylalanine. Oh, yung pinaka-controversial na pronunciation of phenylalanine or phenylalanine. Oh, ikaw, saan ka? Di ba? But to pronounce it correctly, it's really no phenylalanine. Okay? Phenylalanine. Okay? So, this one, what I would like to emphasize in this one is that it is used by the brain to produce your neurotransmitter norepinephrine. Okay. Also, this amino acid uses the active transport channel to cross the blood-brain barrier, and it interferes with the production of serotonin. Later, in the next slide, I'm going to describe more about serotonin because the next amino acid is the precursor no, of serotonin. And then your phenylalanine also is part of this what we call aspartame. So itong aspartame po ha, mag-ingat po tayo dito. Bakit po? Kasi may iba, pag sinabing aspartame, no, baka ang isasagot ay aspartame. Ang amino acid na nandyan ay si aspartate or your aspartic acid. So that is wrong, no? Wrong association. So when we say aspartame po, this one is associated with phenylalanine. Okay? And your aspartame class is a common sweetener used in prepared foods, no, as sugar replacement. Then we have threonine. So hindi pa pala yung next amino acid, no, yung ating serotonin. So yung threonine po, so ito po yung kanyang description. So ito sila, kinuha ko lang po ito sa Bishop na libro. So hindi ko na po siya ibabasa for you. Okay? So you know how to read naman. And hindi ko na kailangan to siya i-elaborate. Or, yeah. Kasi madali lang siya intindihin, no, yung description. For tryptophan, this one, there are some things that I would like to talk about for tryptophan. So for tryptophan, ito yung amino acid na sinasabi ko kanina during phenylalanine, no, sa slide na phenylalanine, na meron po tayong amino acid that is considered as the precursor for serotonin and melatonin, and that is again your tryptophan. So before I proceed, let us first describe these two, no, serotonin and melatonin. So when we say serotonin po, the scientific name of your serotonin, let me type it so that we will have a uniform spelling. So the scientific name for serotonin is 5-hydroxytryptamine. Okay? 5-hydroxytryptamine. Or this one is abbreviated as 5-HT. No? So that is a scientific name for serotonin. And actually, no, if I'm going to describe serotonin, serotonin po is considered as a happy chemical. No? Ay, meron pala tayong ano, no? Meron pala talaga yung totoong sinasabi nating happy pill, no? But this time, natural siya, no, na happy pill. Hindi yung happy pill na tao yung ginamin natin. No? Ay, ikaw yung aking happy pill. Okay, so hindi, no? So in this circumstance or in this scenario, no, natural, okay, no, na nasa loob ng or rather um, natural na biomolecule, no, ang ating kino-consider dito. Okay? So we have tryptophan. So this one is a happy chemical. It is described as a happy chemical because it can stabilize the mood of a person. So yung may mga mood swings dyan, parang bagay sa inyo ang tryptophan. O di kaya may mood swings kayo kasi kulang ng tryptophan. Okay? So anyways, so your serotonin is a happy chemical. It can stabilize the mood. It is also actually responsible for the person feeling happy. Nag-iingon na happy chemical siya. For melatonin naman po, I hope you're still familiar with this one. So remember po, melatonin is a hormone secreted by your pineal gland. So this one plays a role in our sleep-wake cycle. And it is highest during nighttime. So that's for melatonin, okay? So the precursor again for serotonin and melatonin is tryptophan. So what are the other effects of tryptophan in our body? So it is a natural relaxant. So it alleviates insomnia by inducing sleep. No? You can use it to treat migraine headaches. No? And I would also like to emphasize this one. So tryptophan is the heaviest among all the amino acids. Okay? Okay. So that's for tryptophan. So again, for the other bullets, you're just going to read na lang those, okay? Para hindi masyado mataas yung video lecture. Then the next amino acid is valine. So what I would just like to emphasize here is that it can be used by the muscle tissues as an energy source. Then we have alanine. For alanine po, it is a product of the breakdown of your DNA. Okay, so also it is responsible for the transfer of nitrogen from the peripheral tissue to the liver. Then we have asparagine. So this one is said to be the first amino acid to be isolated. So ang pangalan po ng amino acid na ito ay asparagine because it was isolated from asparagus juice. Okay? And also, this amino acid is involved in the processes called amination and transamination. So what is the difference between the two? In amination, remember, it is a process wherein an amine group is introduced to an organic molecule. For transamination naman po, it is a reaction wherein, or it is a process wherein the amino acid is transferred to an alpha keto acid. Usually, the alpha keto acid that we are dealing here is alpha ketoglutarate. If you're going to recall your topic in metabolism, no, sa biochemistry nyo. And also, I would like to let you remember or recall, no, na remember po, amination and transamination, these are processes that occurs before urea cycle, right? Okay, so that's the role of asparagine, no, to convert an amino acid to another via amination and transamination. Next po is your aspartic acid. This one is also known as your aspartate. Okay. So this one is a metabolite in the urea cycle, and it participates in gluconeogenesis. By the way, this is not anymore new to you, gluconeogenesis, because you have already encountered this one in biochemistry, specifically in the metabolism topic, as well as in the carbohydrates topic, last prelims, correct? So what is gluconeogenesis? Gluconeogenesis, it is a metabolic pathway wherein we produce glucose or we synthesize glucose or perhaps pyruvate, no, from non-carbohydrate sources. Okay, so that's for gluconeogenesis. And again, aspartic acid participates in this metabolic pathway. Then we have your cysteine. cysteine. I pronounce it as cysteine, not cysteine, because we have a compound called cysteine as well. So to differentiate the two, that's why I pronounce this amino acid as cysteine. Okay? So the word cysteine here is named after its precursor, cysteine. So, sir, the question now is, sir, paano ako makakabuo ng cysteine galing sa cysteine? So what happens here is that yung cysteine nyo class, yung cysteine, it will travel to your cells. It will go inside your cells. And then as it goes inside the cells, your cysteine will be reduced to 2 cysteine molecules. Okay, so that means for every 1 cysteine molecule that enters the cell, you can produce 2 cysteine molecules. Okay, that is why your cysteine is also known as a half cysteine residue. Half cysteine residue. Okay, because of how it is formed from cysteine, half cysteine residue, cysteine. Okay, so if you can see at the side chain, it is also containing sulfur. So if I'm going to ask you what are the 2 amino acids that contains sulfur in the side chain, your answer would be 2. That would be cysteine, which is the first one, and the second amino acid that contains sulfur is methionine. Okay? Okay. Also, your cysteine is involved in the production of flavors. Okay? So let me delete this one. Now, let's have glutamic acid. So glutamic acid is also known as glutamate. Now, this one can serve as a neurotransmitter. And if there is dysregulation, meaning abnormal yung regulation ng ating glutamic acid, this could lead to this what we call epileptic seizures. Okay? So this one is responsible for the taste umami. Okay? And you can commonly see this amino acid incorporated in this what we call monosodium glutamate. Mas kilala sa tawag na bitsin. Okay? So, no, marami kayong makikitang glutamate or glutamic acid. Okay? Then we have glutamine. Now, glutamine is the most, kung meron tayong heaviest amino acid, meron tayong first amino acid to be isolated. So we have the most abundant amino acid. Meron pa nga tayong simplest later. Simplest amino acid. So this one is the most abundant amino acid in the body. So its role, one of its roles is to regulate the pH of the body, the acid-alkaline balance of the body. Also, if you are doing weightliftings or bodybuilding, so you can use this as a supplement. And the last rule that I would like to emphasize for glutamine is that it transports ammonia to the liver. So why does glutamine need to transport ammonia to the liver? Ano palang meron kay ammonia? Remember po, your ammonia or your NH3 is toxic to the body. So if you have increased levels of ammonia, that is dangerous. It could lead to the patient having a comatose. So that is why ammonia needs to be transported to the liver so that ammonia will be converted to urea through the process or through the pathway called urea cycle. And the amino acid that transports ammonia to the liver para ma-convert siya to urea is your glutamine. Okay? Next po, your glycine. So this one now is the what we call simplest amino acid. Okay? So as you can see, sa side chain pa lang niya, isa lang na element, and that is your hydrogen. And since isa lang po yung element niya sa side chain, and then very basic yung structure ng amino acid, so there is no stereoisomer for this one. I hope you still remember what a stereoisomer is. I'm not going to discuss that again because that is basic chem already. Okay? So, lysine also has a sweet taste. You can also use it as a sweetener, just like your aspartame. And then it also helps in the synthesis of your bile acids. So I hope you still remember examples of your bile acids, di ba? Main bile acids in our body is your folic acid and your, I'm not sure how to pronounce it, yes, but it's deoxycholic acid, no? So quinodeoxycholic, that's one word, and then space, and then acid. Okay, so those are the main bile acids in the body. Okay, and what helps in the synthesis of those bile acids? That is your glycine. Okay, then you have proline. So this one is the precursor of hydroxyproline. No? And then this one is technically not an amino acid, right? No, it is an amino acid. Why? Because if you're going to look at the structural formula of proline, no, you cannot see the amine group here. Rather, you will see the amino group. Okay. Then we have serine. So serine, ito yung masasabi ko na amino acid ng mga matataba. Okay, serine. Why? Because if we're going to look at the description of serine, no, descriptions are related to fats. Huh? Tingnan nyo, serine needed for proper metabolism of fats and fatty acids. No? Serine is highly concentrated in all cell membranes. And sir, saan ang fat dito? Remember, your cell membrane is a phospholipid bilayer. Okay? And then, it is also a component of the protective myelin sheath. And then your myelin sheath also contains fats, right? No? So, no, anong tawag nito? Ano nito ay? Amino acid ng mga matataba, no, of fats. Okay? That's for serine. Then we have tyrosine. For tyrosine po, it is a precursor of your neurotransmitters, epinephrine or epinephrine. It is also a precursor of dopamine. And then also, I would like to emphasize, tyrosine is also a precursor of your T3 and T4. Remember, your T3 and your T4 are your thyroid hormones. No? T3 is also known as thiiodothyronine. T4 is also known as thyroxine. Sir, sa spelling. Search sa Google kung hindi sure sa spelling. So your T3, triiodothyronine, and then your T4, thyroxine, their precursor is tyrosine. Okay? So expect po na if you're going to look at the structure of the thyroid hormones, you will definitely see tyrosine there. But I would like to inform you as early as now, kasi i-discuss din ito sa clinical chemistry 2, no? Pag mating na tayo sa endocrinology, yung ating thyroid hormones po, hindi lang yan siya purely tyrosine, ha? Okay? So, T3 and T4 po, aside from tyrosine, nandiyan pa rin po si iodine. Okay? So, ito yun siya, no? For your thyroid hormones. And as I've mentioned a while ago, no? Bago pa lang tayo nagsimula sa video lecture, there are two new amino acids. But for me, it's not anymore new. No? Kasi po, na-establish ito sila 2002 pa. Okay? So, you have your selenocysteine and your pyrrolysine. So, for your selenocysteine, the abbreviation for this one is SEC. Murag Securities and Exchange Commission, no? SEC. So, this one is encoded by your UGA codon. So, ito yung amino acid ng mga uga. Okay? So, meron tayong amino acid sa mga matataba, no? May amino acid din para sa mga uga. So, sa mga uga dyan na nakikinig, no? This is your amino acid, selenocysteine. Okay? So, this one, its abbreviation is SEC. So, it's actually a selenium analog of cysteine, or rather cysteine, para hindi tayo malito. Selenium analog of cysteine. Okay? So, what do we mean when we say selenium analog? When we say selenium analog po, it means that the sulfur in your cysteine, is replaced by selenium. Okay? So that's the difference between the two. Okay? And also, your selenocysteine is found in some enzymes. No? In your Bishop book, naka-enumerate po doon yung mga enzymes. So the enzymes are your formate dehydrogenase, your glycine reductase, no? And some hydrogenases. By the way, if I'm quite fast, no, in terms of pacing, no, pwede nyo po tawag nito, i-modify or i-edit yung playback speed. Baka kasi medyo mabilis na ako para sa inyo. Okay? So anyways, continuing with the discussion. So aside from selenocysteine, you also have your pyrrolysine. So kung meron tayong amino acid para sa mga matataba, sorry, kung maigo, no? Taba ang pod ko. So same lang, no, ang effect. Okay. So kung meron tayong amino acid para sa mga matataba, meron tayong amino acid para sa mga uga, meron po tayong amino acid din para sa mga uwag, no? Sa mga mahilig mangigat. Okay? Uwag. Okay, so why did I say that? Because your pyrrolysine is encoded by the UAG codon. Okay? So that's for pyrrolysine. And also this amino acid is used by archaea and unicellular organisms. Now, you might wonder, sir, stop codons man po yung ating UAG at saka yung UGA. So how come that they can still encode amino acids? No, they can still be used to encode amino acids. So the answer for that one is in your book. So I'm going to leave that one to you so that you will read your book. Okay, so try to know what is the explanation. Why is it that even though they are stop codons, they are still capable of encoding amino acids? Okay, so that's for our amino acids. By the way, I forgot to edit this number. So this is 21 and then this is 22. 22 amino acids pa lahat. Okay, so we're done discussing the descriptions, no, or the characteristics of each amino acid. Let us now proceed to the metabolism of your amino acids. Okay, so we're done again. Sorry for that. We're done with our amino acids. Let's proceed with the metabolism. Okay, so how do our amino acids metabolize? So first, remember our amino acids, saan sila makikita? Especially yung mga essential amino acids sa pagkain. So from the food, no? Hindi pa yan sila amino acids agad, no? Ang makukuha natin sa food is yung protein pa, no? Complex pa na version ng ating amino acids. So kunin natin yung protein from the food, of course, via ingestion. So once you ingest the protein, what will happen? Proteins will be initially digested by the enzymes pepsin and trypsin, correct? So once it will be degraded, once the proteins are degraded by your pepsin and trypsin, you will now have your amino acids. These amino acids now will be absorbed in the intestine and it will become part of the amino acid pool. Yun, class. Itong tinatawag nating amino acid pool, dito kinukuha yung mga amino acids na gagamitin later sa translation. Okay? Okay. So, once the amino acid is degraded, no, instead of utilizing the amino acid in making a protein, once the body opts to degrade the amino acid, the product of the degradation now will vary depending if the amino acid is said to be ketogenic or glucogenic. So what's the difference between the two? If we say ketogenic amino acid, itong mga naka-color blue, okay, so itong mga amino acids po na ito, when they are degraded, no, ang product po nila ay either acetoacetyl coenzyme A or acetyl coenzyme A. That is acetoacetyl coenzyme A, that could be converted to acetyl coenzyme A, or they could be placed in the metabolic pathway that we call ketogenesis. Okay? So, that's for acetyl coenzyme A. For acetyl coenzyme A naman po, pwede siyang ipasok papunta sa ketogenesis. If you still recall, again, your metabolism na topic sa biochem. Or, this acetyl coenzyme A will be, this acetyl coenzyme A will be incorporated, or rather will be allowed to enter the citric acid cycle. Okay? So, yan po para sa ating ketogenic amino acids. For glucogenic naman po na amino acids, ito mga naka-color pink, yung glucogenic amino acids natin, when they are degraded, they can generate precursors of glucose, no, like your pyruvate. Okay? And then from pyruvate, pwede ka nang magkaroon ng gluconeogenesis, o di kaya from pyruvate, gagawa siya ng acetyl coenzyme A and then ipapasok siya sa citric acid cycle. Or other glucogenic amino acids naman po, instead na pyruvate, ang gagawin po ay product nila magiging citric acid cycle intermediate, and that citric acid cycle intermediate now will be part again of the citric acid cycle. Okay, so if you can see here, this is now how the ketogenic and glucogenic amino acids, no, are metabolized. Okay. So, another thing that I would like to talk about regarding the metabolism of amino acids is that, diba, there is also a process wherein yung ating amino acid, yung kanyang amino group, no, yung amino group ng amino acid natin, tatanggalin. Okay? Diba, merong ganon sa metabolism. No, pag pinag-uusapan natin ang metabolism ng amino acids. So, kapag po tinatanggal yung amino group galing sa amino acid, that is done through what? Through oxidative deamination or transamination. Okay? So, I hope you're still familiar with this, ha? No, kung hindi na, sus, balikan ng biochem, please. Okay? So, again, when talking about metabolism of amino acids, there is also, no, a process wherein the amino group from the amino acid is removed through oxidative deamination or transamination. And what is the result? No? After deamination or transamination, the result will be a keto acid. Usually that is your alpha ketoglutarate. No? Keto acid, and tanggalan mo man ang amino group. Yung amino group magiging ammonia. No? That is your NH3. So dalawa yung products natin after deamination or transamination of an amino acid. You have your keto acid and your ammonia. Okay? The keto acid there, class, will enter the common metabolic pathway. Okay? Whereas yung inyong ammonia, that is toxic kasi to the body, diba? I've mentioned a while ago. So that ammonia should be converted to less toxic form, and that is your urea. And how do we convert ammonia to urea? Diyan na po papasok yung ating urea cycle. Okay? Okay, so that's for the metabolism of our amino acids. Okay, so now let us proceed. No? Kasi yun, biochemistry pa yun. So let's proceed with the clinical applications for your amino acids. So you have this what we call amino acidopathies. So when we say amino acidopathies po, these are rare, inherited disorders, meaning this involves genetic defects. Okay, so rare inherited disorders of amino acid metabolism. Okay, and usually your amino acidopathies are caused by either what? An enzyme defect, or an enzyme deficiency, or an abnormality in the membrane transport system. Again, I would like to clarify, no, na itong problema natin sa enzyme or sa membrane transport system is again brought about by a genetic defect. Okay? So, again, ang gusto ko lang po sabihin, ang amino acidopathies, no, nandyan sila, they are caused by a problem with an enzyme or a problem with the membrane transport system brought about by a genetic defect. Okay? So, let us now discuss the amino acidopathies. Please, don't forget this topic. Why? Because your amino acidopathies will also again be repeated in your UBF next semester. No? Kasi yung ating amino acidopathies po kasi, no? In terms of diagnosis, no, ginagamit rin natin yung specimen na urine, no, aside from blood. Okay? So please don't forget about this one. So let's begin with the first amino acidopathy, and that is your PKU or your phenylketonuria. Okay? So this is an autosomal recessive disorder. Okay? So in other words, you're going to inherit two mutated genes, no? One from each parent. And I guess you're familiar with the term autosomal recessive already because you had already your cytogenetics, na? Yung favorite cytogen. Okay? So in this amino acidopathy, the problem is with an enzyme. No? So because of the autosomal recessive disorder, no, there is an absence now of an enzyme called phenylalanine hydroxylase. Okay? So, what is the role pala of this enzyme? The role of this enzyme class is to convert phenylalanine to tyrosine. Okay? So, since in your PKU, wala po itong phenylalanine hydroxylase, so hindi mako-convert yung phenylalanine mo to tyrosine. This is why what happens is that magkakaroon ng accumulation or build-up of your phenylalanine. Okay? And then, I would also like to inform you, no, na aside from the build-up of phenylalanine, magkakaroon ka rin ng build-up ng tawag nating metabolites of phenylalanine. You will encounter them in the next slide. Ano ba yung mga metabolites of phenylalanine? Yun ha? Pag meron kang PKU, absent yung enzyme na ito, so yung phenylalanine mo, hindi mako-convert to tyrosine, so there will be build-up or accumulation of phenylalanine. Then also a build-up of the metabolites of phenylalanine. So of course, if there is accumulation in the blood, no? If there is accumulation of the blood, ang tawag natin sa condition is hyperphenylalaninemia. Hala sir, na-confuse ako. Ang pinag-uusapan natin is phenylketonuria. Bakit meron na namang hyperphenylalanemia? Ganito po kasi yan. Kapag po ang pinag-uusapan lang natin muna ay yung accumulation or increase of the levels of phenylalanine in the blood, that is what we call hyperphenylalanemia. Okay? Pero po, meron kasi tayong tinatawag na renal threshold. Ano itong renal threshold? Remember, yung kidney po natin, di ba, renal, kidney. Yung kidney po natin, siya yung nag-filter, correct, ng ating dugo. However, meron rin pong limit yung kanyang kayang ma-filter. So ngayon, dito sa hyperphenylalanemia, marami ka masyadong phenylalanine. So since marami ka masyadong phenylalanine, hindi makakayanan ng kidney mo na i-filter lahat. So ano mangyayari? Since hindi makaya ng kidney na i-filter lahat, so yung phenylalanine magiging present ngayon sa urine. The moment that your phenylalanine is present in the urine, the condition now is called phenylketonuria. Okay, you have the suffix here, urea naman. Okay, so that's the difference between the two. This one refers to a condition wherein there is an increased level of phenylalanine in the blood. This one now is the presence of phenylalanine in the urine. Okay? Okay. Okay. Saan na ako? Let me delete first this one. Okay. So what are the complications? Okay. So if a person, like a newborn or a baby has phenylketonuria, then expect po na meron siyang mental retardation. No? And aside from mental retardation, expect po na meron din siyang microcephaly. Microcephaly. Okay? So, if yung mama, no, if the mother has phenylketonuria, actually, you can prevent that one. No? For example, what do I mean by this, no, na circumstance? For example, ang mama merong phenylketonuria, ang baby wala. So, maagapan mo na ma-experience ng baby yung effects ng phenylketonuria. How? No? Yung mama, i-make sure mo lang na yung mama mag-undergo siya na tinatawag nating phenylalanine restricted diet. Okay? So, ipa-diet mo si mama. Okay? Para yung kanyang baby hindi mag-suffer ng mental retardation and microcephaly. No? So, maagapan, no? If ang bata normal and then yung mama ang may PKU. Ang mahirap talaga dyan is if ang bata mismo ang may PKU. Okay? Yan ang mahirap. Okay? Also, a while ago, I have mentioned no, na kapag sinasabi natin PKU, is an increased no, level of phenylalanine and its metabolites sa dugo resulting to its presence in the urine. So ito po yung mga metabolites na sinasabi ko kanina. So you have your phenylpyruvate, which is a result of the deamination of your phenylalanine. You have your phenylacetic acid, which is the result of the decarboxylation and oxidation of your phenylpyruvate. And you have your phenylacetylglutamine, which is a glutamine conjugate of your phenylacetate. So these one are the metabolites of your phenylalanine. And these metabolites are also increased in your PKU. Okay? Next, we have the methods of determination. So how do we diagnose if a person has PKU? So first, we have this what we call Guthrie test or the Guthrie bacterial inhibition assay. So anong ginagawa po dito? Now, what we do in this assay is that we try to use the ability of phenylalanine that could be present in the specimen to facilitate the bacterial growth in a culture medium with an inhibitor. So the bacteria that we are growing in the culture medium here is your Bacillus subtilis. Uy, bakte. Okay? So, your Bacillus subtilis. So, ang inhibitor naman po na ilalagay natin sa culture medium or sa agar plate ay si beta-2-thioenolalanine. Okay? So bakit siya tinawag na inhibitor? Because this one will antagonize the growth of your Bacillus subtilis. Now, para mas madali ninyo ma-imagine class, the concept of the Guthrie bacterial inhibition assay is quite similar with your antibiotic susceptibility testing. Why? Because what we do in your Guthrie test is meron kayong filter paper disc. So, parang yun siya yung antibiotic disc sa inyong bakte. Meron kayong filter paper disc that's impregnated with the blood no, from the newborn or from the infant. And then that disc will be it will be placed no, sa ating agar plate na merong inhibitor na beta-2-thioenolalanine. Ngayon, to interpret the result, if may bacterial growth around the filter paper disc, that means that the patient is positive for PKU. If walang bacterial growth na nag-surround sa disc, that means negative for PKU. So para mas ma-imagine pa, we have here a picture. Okay? So this one is the culture medium. Okay? So this one is the filter paper disc impregnated with the blood of the infant. Ng red. And then, kung mapapansin nyo po, may parang halo na nakasurround sa filter paper disc. So itong halo na ito, that implies na merong bacterial growth. Nag-grow si Bacillus subtilis. Okay? Kapag wala naman pong growth, wala siyang halo. So, medyo opposite yung description ngayon with your antibiotic susceptibility testing. Kaya di ba, sa inyong sensitivity testing, dapat clear ang nakasurround sa disc. And that's what we call the zone of inhibition. Dito naman sa Guthrie test, opposite siya. Instead the clear, ang clear ay yung surroundings and then yung magiging surrounding naman ng ating filter paper disc, yun naman yung magiging cloudy. Okay? So, ang positive natin dito ay bacterial growth. And that is seen through this halo. Yan yung kaibahan ngayon ng sensitivity testing nyo sa bakte dito, sa Guthrie test. Okay? So, Ano ba ang rationale? No? Bakit nag-grow si subtilis kapag may PKU ang ating pasyente? Kasi po, yung phenylalanine, no, yung ating phenylalanine na present sa pasyente na may PKU, the phenylalanine there will neutralize the inhibitor in the culture medium. In other words, your phenylalanine will neutralize the beta-2 thioalanine. And since in-neutralize niya ito na inhibitor, wala nang kakalaban sa growth ni Bacillus subtilis. Okay? So, kaya po makaka-observe ka ng bacterial growth around the disc. Okay? Okay. So, in terms of sensitivity of this test, no? If the patient, no, meron na siyang phenylalanine blood level na 3, or rather more than 3 to 4 milligrams per deciliter, that's the time that you can observe, no, for the halo. No? You can start observe for the halo. Now, aside from the Guthrie test, another method of diagnosing your PKU is your microfluorometric assay. So, what do we do here? Okay? So, meron pa rin po tayong filter paper na gagamitin, no? And that filter paper must be pre-treated with trichloroacetic acid. By the way, the filter paper already contains the specimen. Okay? So, nandiyan na po yung specimen natin sa filter paper. So the filter paper specimen, no, with the specimen, is pre-treated with trichloroacetic acid or your TCA. No? And then, we will allow the extract, no? Kasi gagamit kasi tayo ng extract dito sa microfluorometric assay. So we will allow also the extract here to react with the microtiter plate, no? And that microtiter plate contains a mixture of phenylalanine, or rather not phenylalanine. The microtiter plate originally contains your ninhydrin, your succinate, your lucil-alanine, no? And also your copper tartrate. So, we will allow the extract, no? Meron tayong extract na ginagamit sa microfluorometric assay. We will allow the extract to react in the microtiter plate that contains your ninhydrin, your succinate, your lucil-alanine. Lucil-alanine is one word. And your copper tartrate. Okay? So, we will allow the extract to react with that one. And then, nandyan din po yung ating, dadagdag din po natin dyan yung ating filter paper na pre-treated with your TCA. And then, you're going to observe for fluorescence. Okay? So, when we are already about to observe for fluorescence, you're going to use the excitation and emission wavelengths of 360 and 530 nanometers respectively. So ang excitation wavelength na gagamitin ay 360 nanometers. Ang emission wavelength na gagamitin ay 530 nanometers. Okay? Okay. So let's proceed now with the other methods of detecting or diagnosing your PKU. So the reference method, when we say reference method, this is not the standard method for diagnosing your PKU. That is your HPLC. And then, if your specimen naman po is urine, and you are already diagnosed with PKU, but you want to monitor the levels of phenylalanine or its metabolites in your urine, no, you can use the reagent strip test. The reagent strip procedure here is quite the same with the reagent strip procedure that you did in your biochemistry, no, doon sa inyong urinalysis. So, i-dip lang din siya sa urine and then you observe for a color change. So, the principle of the urine test in your, what they call this, the principle of the urine test in your PKU, for your PKU rather, is that you will allow, no, the specimen or the sample to react with ferric chloride with phenylpyruvic acid in the urine. The phenylpyruvic acid here, again, is the possible analyte that could be present in the urine of the patient. So you will allow, no, again, you will allow the ferric chloride in the strip react with the possible, no, phenylpyruvic acid in the urine. By the way, why do I say possible? Baka kasi, no, baka lang kasi hindi masyado ganun karami yung phenylpyruvic acid sa isang pasyente. So, baka magkaroon ka ng false negative. No? Baka malay mo, sa isang pasyente, mag-iba-iba kasi yan minsan. So, baka sa isang pasyente, mas marami sa kanya yung other metabolites ng ating phenylalanine. So, hindi masyado ma-observe yung color change sa strip test. Okay? So, yun lang. No? Yun lang yung isa sa mga possibilities. Pero if umabot siya sa sensitivity ng strip test, no, you will really definitely see a color change. No? The phenylpyruvic acid will react to your ferric chloride, and then you will really observe the color change there. No? And the color change that you will observe is green. Okay? Then another amino acidopathy is your tyrosinemia. By the way, before I proceed with tyrosinemia, I would also like to tell you, no, or I would also like to remind you na yung ating PKU is one of the diseases, no, that is tested for, or rather in newborn screening here in the Philippines. Okay, no? So, additional information lang po yun siya. Now, let's proceed with the second one, your tyrosinemia. So, this one is characterized by the excretion of tyrosine and its metabolites or catabolites in the urine. Okay? So, there are three types of tyrosinemia. You have type 1, type 2, type 3, depending now on the enzyme concerned. No? If the enzyme that is problematic, no, is your FAA hydrolase, then that is type 1. And then if it is 4-hydroxyphenylpyruvic acid oxidase, it's type 2. And then for type 3, you have your tyrosine aminotransferase. So between the, no, not between, among the three, what is the most severe? The most severe na, no? The most severe form of tyrosinemia is type 1. Because actually, if you're going to go back again to your book, try nyo, go to page 209, no? Babasahin nyo po yung symptoms ng type 1. Grabe po talaga yung symptoms niya, no? No, actually, one of the possible symptoms of type 1 is failure to thrive. No? Kaya po, among the three types of tyrosinemia, the most severe form is your type 1. Okay? So complications or other clinical manifestations of patients with tyrosinemia, they could have liver damage. So pwede sila magkaroon ng cirrhosis. Cirrhosis, that is scarring of the liver tissue. So napapalitan yung hepatocytes natin with fibers, no? Scars. Okay? Also, highly possible na patients with tyrosinemia magkakaroon ng liver cancer. Okay? So, ito po, pinapakita lang po yung pathway on how phenylalanine is metabolized. No? Phenylalanine is metabolized to tyrosine. So, again, if ito yung problema na enzyme, you have your PKU. Okay? If dito naman yung problema mo ng mga enzymes, that is now your tyrosinemia. So, the type now will depend on the enzyme that is problematic. Okay? Then we proceed to your alkaptonuria. For your alkaptonuria, it is described as an autosomal recessive disorder as well. So, ang problema mo na gene dito is what we call the HGB. Nasa book yan. HGB gene. Okay? So, yun po yung may problema natin dito, no, ng gene. And then, enzyme pa rin po, no, yung ating concern dito. So the enzyme of concern here is your hemophentisate oxidase. So patients with alkaptonuria lack this one. So what happens is that because of the lack of this enzyme, there will be now an accumulation, no? There will be now an accumulation of your HGA or your homogentisic acid. Okay? So once this chemical accumulates na yung homogentisic acid natin, homogentisic, that's one word, then space and acid, that could cause a generalized pigmentation in our tissues. And that condition now is what we call ochronosis. Ochronosis. And then also, one of, or rather another clinical manifestation of a patient with alkaptonuria is that nagda-darken po yung kanilang ihi. No? No, nagda-darken yung kanilang ihi once it is mixed or once it is exposed to air. Okay? So, parehas ang picture na ito. No? Kapag ito binuksan mo, magiging dark yung urine. Okay? So, yun siya. And then, this one, ito yung sinasabi natin na magkakaroon din ng pigmentations yung tissues. So, this is ochronosis. Okay? So, saan po ba makikita itong enzyme na ito? No, tingnan natin sa metabolic pathway. So, dito po makikita yung ating homogentisic acid oxidase. So, ito yung problema natin sa alkaptonuria. So, since ito yung problema, so magkakaroon ka ng accumulation ngayon ng homogentisic acid, again, leading to your what we call ochronosis, and then leading to your urine becoming dark when exposed to the air. Okay? Next po is your MSUD or your maple syrup urine disease. Okay. Okay, so for your maple syrup urine disease, this one, same with your PKU, this one is also tested in your newborn screening. This one is also an autosomal recessive genetic disorder. So, amuyin nyo yung mga kasama nyo sa bahay, no? Baka merong mga hininga, may mga ma-observe kayo na ang hininga nila parang burnt sugar odor, or di kaya, no, kanang mga dili mamukbo sa CR, no? Kaya, nangihi, wala na mukbo. Pag amoy nimo, uy, burnt sugar odor. O di kaya, pagpawisan, gikan sa gawas, no, nag-exercise, nag-jogging, pag-abot sa imuhang kauban sa balay, no, amoy burnt sugar odor. So, oy, baka MSUD, okay? So, maple syrup urine disease. So, what is the characteristic of this disease now? This one, you have a lacking of 3 branch chain alpha, or rather, not lacking 3, but rather you are lacking 1 enzyme. That's your branch chain alpha keto acid decarboxylase. And this will now block the metabolism of the 3 amino acids. 3. Okay. So you have your leucine, isoleucine, and valine. Okay. So again, magkakaproblema ka sa metabolism ng 3 amino acids na ito dahil you are lacking with this enzyme. Okay? So, possible complications when you have this disease: lethargy, no? Failure to thrive. Ito yung pinaka-worst talaga. You have muscle rigidity, respiratory irregularities, mental retardation, convulsions, acidosis. When you see acidosis, that means the pH of your blood decreased. Mas nagiging acidic siya. And possibly also hypoglycemia. Okay? So, how do we diagnose a person with this disease? You can do modified Guthrie test. So, why is it said to be modified? The procedure, no, is the same with the Guthrie test a while ago in your PKU. Difference lang po is the inhibitor, no, that is incorporated in the culture media or in medium. So in here, the culture medium is incorporated with the inhibitor for azalucine. Okay? Now, same also with your PKU, you can also use the microfluorometric assay for your diagnosis of MSUD. So, ang difference lang din po, of course, first, ang difference ng microfluorometric assay ng MSUD with your PKU, first po, yung ating filter paper, no? That will be, instead of pre-treating it with PCA, you're going to pre-treat this one with a mixture of methanol and acetone. Okay? So methanol and acetone. And then the extract, instead of mixing it with ninhydrin, no, with copper tartrate, no, you're going to add leucine dehydrogenase. Okay? And then, in terms of the observance of the fluorescence, you're going to use an excitation wavelength of 360 nanometers, that's 360 nanometers, and then the emission wavelength would be 450 nanometers, 450. Okay? So yun siya. And then in terms of sensitivity, once the result is more than 4 milligrams per deciliter, okay? So makikita niyo na yung fluorescence. No? Klaro na masyado yung fluorescence. Okay? So that's for the microfluorometric assay for your MSUD. Okay. Then other amino acidopathies, you also have isovaleric acidemia. This one again is an autosomal recessive disorder. Again, actually both. No? Si isovaleric acidemia and your homocystinuria, both of which are autosomal recessive disorder. Both of which may problema sa enzymes. Si isovaleric acidemia, problema niya ay si isovaleryl coenzyme A dehydrogenase. Then, for diagnosis, if gagamit ka ng urine specimen, maaamoy mo no, na meron siyang sweaty feet. I'm not sure about this one kasi wala ko siya na personally encounter sa lab. Pero theoretically, no, meron daw sweaty feet odor yung amoy ng ihi ng taong may isovaleric acidemia. Okay? Aside from smelling the urine, no, you can also you should hindi man reliable na smell na ang basehan sa diagnosis. So you should also perform what we call tandem mass spectroscopy or your chromatography. Okay? So yun siya for your isovaleric acidemia. For your homocystinuria naman po, homocystinuria. So mataas or increase yung level ng ating homocysteine. Kasi po, there is a problem with the enzyme cystathione beta synthase. This enzyme po kasi is involved in the metabolism of homocysteine or the conversion of homocysteine to cysteine. Okay? So, you have a problem with this enzyme. So, magkakaroon ka ngayon ng accumulation of your homocysteine. So, para mas makita nyo, so ito po siya. No? So, from methionine to cysteine ang pinapakita dito. So, may problem daw sa enzyme na cystathionine beta synthase. So, hindi mako-convert si homocysteine to cysteine. Kaya mag-accumulate ang substance na ito. Okay? Okay. So, for diagnosis of your homocystinuria, there are two. No? You can use your cyanide nitroprusside urine spot test and your silver nitroprusside test. Both of which, ang specimen dito ay urine. Okay? So, in your cyanide nitroprusside urine spot test, what we do here is yung cysteine and homocysteine mo that could be present in the urine sample or that is present in the urine sample, they will be reduced by sodium cyanide to free thiol. And then itong free thiol mo, this one will be reacted to your sodium nitroprusside and then you will observe for a red-purple color. On the other hand, for your silver nitroprusside test, this one, for homocysteine lang siya, what we do here is we allow silver nitrate to reduce your homocysteine and then you observe for a reddish color. Okay? So, possible complications for your homocystinuria are the following. No? Then we have citrullinemia. Malapit na tayo matapos class, ating amino acidopathies. Okay? Malapit na kayong mag-break. Okay. So for your citrullinemia, this one results from, again, a problem with an enzyme. No? There are two types. You have type 1 and type 2. For type 1, ito yung mismo may problem sa enzyme. So, ang problem niya is yung enzyme na AS. Libot, AS. So, your arginino, careful dito ha, baka makulangan, no? Arginino succinic acid synthetase. So, asan yan si AS makikita? No? Makikita po ito sa urea cycle. No? Kasi nga, di ba, citrullinemia is brought about by problems in enzymes found in the urea cycle. So, your type 1 lack the enzyme AS. Asa na si AS? Dito siya. Okay? So you have an enzyme deficiency of your AS. That is why citrulline will accumulate. Okay, hindi man siya ma-metabolize to argininosuccinate. Okay? Dito naman po kay type 2, this one is, by the way, before I proceed to type 2, I also almost forgot, for your type 1, tama, nasabi ko na pala kanina na there is build up of your citrulline. For your type 2, this one is caused by a mutation of a gene, no, that provides instruction for making the protein citrin. Itong citrin po, ano po ito siya? Citrin actually helps transport molecules inside cells that are used, okay? So, si citrin po, they transport molecules na ginagamit ng cells natin to produce or to break down simple sugars, to produce proteins and nucleotides, and even those molecules that are used in the urea cycle. So, since walang naga-code, or rather, since mutated yung gene na gumagawa ng citrin, in other words, makukulangan si urea cycle ng mga essential molecules para makaproceed siya. Okay? Kaya po, maapektuhan yung urea cycle. Okay? So, yan po ang type 2. Yan po yung cause ng type 2 citrullinemia. Okay? And then these are the complications. So, ito ang mahirap. High ammonia levels and mental retardation. Itong high ammonia levels, mahirap ito. Kasi, again, sabi ko kanina, pwede yan mag-lead to comatose. Okay? So, bakit tataas yung ammonia levels? Kasi nga, di ba, may problema sa urea cycle. It is through the urea cycle baya na mako-convert si ammonia to a less toxic form, which is urea. Okay? Then we also have this what we call argininosuccinic aciduria. So this one, also related din ito siya sa enzyme deficiency sa urea cycle. So kung kanina sa type 1, ang problema ay si AS, no? Sa ating argininosuccinic aciduria, ang deficient naman po ay si ASL. Okay, muna mag-omegal, no? ASL. Okay? So, ASL stands for argininosuccinate liase. Okay? So, since this one is deficient, so accumulate si arginine. And the complications will still be the same with your citrullinemia. Okay? Then we have cystinuria, the last amino acidopathy. Yay! Okay, so cystinuria, again, this is an autosomal recessive genetic disorder. So there is a defect here. This time, we're not anymore having a problem with an enzyme, but this time we're having a problem now with an amino acid transport system. Okay, so what happens in cystinuria is that there is increased urinary excretion of cysteine. Why? Cysteine, not cysteine. So we have an increased urinary excretion of cysteine because there is an inadequate reabsorption of cysteine during the filtering process in the kidneys. No? And since may increased urinary excretion of cysteine, so magiging concentrated masyado yung cysteine sa ihi mo. And remember, cysteine is insoluble. So they will precipitate in the tubules of your kidney, and that could now lead to this what we call urinary calculi. Or ibig sabihin, magkakaroon ng kidney stones. Okay? So, si cystinuria, no, yan ang main problem natin sa kanya, no? Kidney stones. Okay? Now, for the last slide of this video lecture, of this part of the video lecture. So this is just a summary of the odors, possible odors, no? And then also the possible causes of those odors. Okay? So, take note lang po yung specimen natin dito is urine. So for example, nag-urinalysis ka, nakaamoy ka ng mousy odor, then that is most probably PKU. Again, I use the term most probably because we have more accurate methods for this one compared sa amoy-amoyin mo lang ang ihi. Okay? So you study this one on your own na lang. Okay? So that is the end for our amino acids. In the next video lecture, we will now discuss proteins. Okay, so that is all for amino acids. Thank you, and see you in the next video lecture, which is proteins.